Welcome! Today, we explore a critical molecular sentinel on the surface of many cells, the death receptor 5, or DR5, also known as trail R2. This receptor plays a pivotal role in initiating programmed cell death or apoptosis. Its unique expression pattern and function make it a fascinating and promising target for treating diseases like cancer, fibrosis, and autoimmune disorders. Let's delve into the world of DR5. We will dive into its abstract expression distribution, structure, biological functions, and signaling pathways and targeting DR5 for therapy. DR5 is a member of the tumor necrosis factor receptor superfamily. It sits on the cell membrane, primarily functioning as a death switch. When activated by its specific signal, it initiates a cascade leading to the cell's self-destruction. Because of this powerful function, scientists are intensely studying how to control DR5 activity either by activating it to kill harmful cells, like tumors, or blocking it to protect healthy tissues in certain diseases. Here's what makes DR5 so interesting for targeted therapy. In most healthy tissues, DR5 is present but at very low levels. However, in a wide range of cancer cells including bone sarcomas, blood cancers, and solid tumors like those in the breast and pancreas, DR5 expression is frequently turned up significantly. This difference between low in normal and high in tumor provides a potential window for selectively targeting cancer cells. Zooming into its structure, DR5 is a single-pass transmembrane protein. Outside the cell, its extracellular domain binds to activating signals. A special motif in its transmembrane helix allows two DR5 molecules to pair up. Inside the cell, the crucial death domain is the engine room for initiating the death signal. Importantly, DR5 can self-assemble into clusters, which is key for amplifying the signal to effectively trigger cell death. How does DR5 work? Its classic job is to trigger apoptosis. When its natural ligand trail or an activating antibody binds, DR5 proteins cluster. This recruits an adapter protein called FAD, forming the death-inducing signaling complex or DISC. The DISC then activates an enzyme called caspase-8, launching the irreversible cell death program. However, if this apoptosis pathway is blocked, DR5 signaling can sometimes switch to non-death outcomes, like promoting inflammation or even a different type of cell death called necroptosis. The main therapeutic strategy is using DR5 agonists, molecules that turn DR5 on. The idea is to deliver these activators specifically to diseases where cells overexpress DR5, like cancers or fibrotic tissues, to selectively induce their death. Examples include tigatuzumab, an antibody drug in trials for cancer, and TLI-012, a designed trail protein being tested to kill fibrosis-driving cells. To make therapies more precise and powerful, next-generation agents are being developed. Take BY905711, a bispecific antibody. It grabs two things at once, DR5, and another tumor marker called CDH17. It only strongly activates DR5 when both are present on the same cell, potentially reducing side effects. Another approach is the antibody-drug conjugate, like RC248. Here, the anti-DR5 antibody acts as a homing missile, delivering a potent chemotherapy drug directly into the cancer cell, offering a targeted double hit. More target sharing. See you next time.